close your eyes for a moment. Imagine being held still, bright lights above you, a chemical dropped into your eyes to see if it burns. That's not a horror movie. For decades, that's been standard laboratory testing. But what if science no longer needed to do this? What if we could grow real human skin in a lab, alive, responsive, biologically accurate, and test products without harming a single animal? This isn't a future concept. It's happening right now, and it may become the technology that ends animal testing forever. Over 100 million animals are used in laboratory testing worldwide each year. Yet more than 90% of drugs that pass animal trials fail in humans. So what if the system we relied on for decades isn't just controversial but outdated? Today, scientists are building something radically different. They're printing human skin in laboratories. And this breakthrough could make animal testing not only unnecessary, but scientifically obsolete. The future of medicine may not involve cages at all. It may involve printers. Every year, millions of animals are used in laboratories, not for survival, not for food, but for testing. Rabbits, mice, guinea pigs, used to determine whether your shampoo burns, your lotion irritates, or your medication heals. But what if I told you there's a future where science doesn't need animals to test safety? A future where human skin can be printed, layer by layer, in a laboratory, alive, responsive, biologically functional. This isn't science fiction. It's happening now, and it may permanently change how we test medicine, cosmetics, and chemicals forever. This is the rise of 3D printed human skin. Animal testing has been a cornerstone of biomedical research for decades. Organizations like Food and Drug Administration and European Medicines Agency have historically required animal data before human trials begin. The logic? Safety. But here's the problem. Animals are not humans. A drug that works in mice may fail in people. A chemical that appears safe in rabbits may irritate human skin. In fact, studies show that over 90% of drugs that pass animal testing fail in human clinical trials. Why? Because biological systems are deeply complex, and species differences matter. Skin thickness, immune response, metabolism, even the way cells communicate. A rabbit's skin is not your skin, and ethical concerns are growing louder. The European Union banned animal testing for cosmetics in 2013. Countries like India and Israel followed. Consumers are demanding cruelty-free science. But here's the catch. If you remove animal testing, you need something better, something human, something reliable. That's where 3D bioprinting enters the story. You've seen 3D printers create plastic objects, toys, prototypes, even houses. Now imagine replacing plastic with living cells. That's bioprinting. Instead of ink, scientists use bioink, a mixture of living human cells and supported biomaterials. These cells are carefully layered, just like real skin, because real skin isn't flat. It has structure, the outer epidermis, the deeper dermis, collagen fibers, blood vessel networks. Researchers at places like Wake Forest Institute for Regenerative Medicine and companies such as Organ Novo have developed methods to print multi-layer human tissue that mimics natural skin. Some models even include immune cells. Others incorporate hair follicles or pigmentation cells. And here's what makes it revolutionary. It's human, not simulated, not synthetic plastic, but living tissue grown from human cells. This means researchers can test products on skin that behaves like yours. It reacts to irritation. It metabolizes compounds. It mounts immune responses. And because it's grown in a lab, it can be standardized, consistent, reproducible, controlled. No variability between species. No ethical dilemmas involving animals. But the implications go even further. Step 1. Cell sourcing. Scientists obtain human skin cells, often donated from surgical procedures or biopsies. These cells are expanded in culture. Millions become billions. Step 2. Bioink preparation. Cells are mixed into hydrogels, materials that mimic the extracellular matrix of real tissue. Think of it as scaffolding, a soft, supportive structure that allows cells to attach and grow. Step 3. precision printing. Using computer-guided systems, printers deposit layers of bioink in micrometer precision, layer by layer, top to bottom, epidermal cells first, dermal cells beneath, sometimes vascular channels in between. Step 4. Maturation. Printed tissue is incubated under controlled temperature and nutrient conditions. Over days to weeks, cells organize. They communicate. They form tight junctions. They produce proteins like collagen and elastin, and eventually it becomes functional skin. Researchers can then apply chemicals, UV radiation, cosmetics, or pharmaceuticals, and measure inflammation markers, cell viability, barrier integrity, gene expression changes, data that is directly human relevant. This is already being used in cosmetics testing and toxicology screening, and regulatory agencies are beginning to pay attention. In fact, the United States Environmental Protection Agency has announced plans to reduce animal testing significantly in the coming years. Why? Because alternative methods like bioprinted tissues are becoming scientifically superior. Here's something most people don't realize. Animal models are predictive, not definitive. A mouse's immune system reacts differently than a human's. A rat's liver metabolizes drugs differently. And skin? It's one of the most species-specific organs in the body. Human skin is thicker than rodent skin. It has different lipid composition, different microbiome interactions. Bioprinted human skin eliminates that translation problem. It removes the guesswork. Instead of asking, will this work in humans? Researchers can test directly on human tissue models. This doesn't just reduce cruelty. It increases precision. It lowers costs long-term. It speeds up drug development and potentially saves lives by identifying failures earlier. There's another major benefit: customization. Scientists can print skin from specific genetic backgrounds. Imagine testing a drug on skin derived from patients with eczema or psoriasis or rare genetic skin disorders. That level of personalization is impossible with animal models. This is where regenerative medicine meets precision medicine. But let's be clear, 3D printed skin is not yet perfect. It lacks full vascular systems in many models. Long-term immune interactions are still being studied. Whole body responses can't be replicated with skin alone. And regulatory frameworks evolve slowly. Before agencies like the Food and Drug Administration fully replace animal requirements, they require extensive validation data. Science moves carefully, for good reason. Yet, momentum is building. Advancements in organ-on-a-chip systems, integration with microfluidic blood flow, even combining printed skin with printed liver tissue for systemic testing. The goal? A fully human, lab-based testing ecosystem. No cages, no animal sacrifice, just human-relevant science. Now imagine the next decade. Cosmetics tested entirely on printed human skin. Burn victims treated with custom-printed grafts. Drug reactions predicted before the first human volunteer ever swallows a pill. Bioprinted tissues connected together, skin, liver, heart, forming miniature human systems on a chip. This is not the end of science. It's the evolution of it, from crude approximations to precision biology, from animal proxies to human reality. The question is no longer if this technology will grow. The question is how fast. Because once we can print living tissue, we change not just testing, we change medicine itself. And perhaps one day, animal testing will be something we look back on as a necessary but outdated chapter in scientific history, replaced by something more humane, more accurate, and profoundly human.